In the next section, we'll describe the physiology of skeletal muscle. In this section, we'll talk about muscle physiology. At the top left of this figure, one can see the neuromuscular junction. At the neuromuscular junction, we have a presynaptic axon coming from an alpha motor neuron, which innervates the muscle fiber. When the presynaptic axon has an action potential traveling down it, it will induce the influx of calcium at that presynaptic synaptic terminal, and that will cause the release of acetylcholine from stored synaptic vesicles. Acetylcholine will bind to a nicotinic cholinergic receptor, which will open up sodium channels and allow sodium to rush into the cell to depolarize it, and that will cause an action potential to occur on the membrane of the skeletal muscle cell. That action potential will cause contraction to occur via, to a certain degree, the influx of calcium from the extracellular space, as well as the release of calcium from intracellular stores. The mechanism of contraction will be the focus of the next several slides. Shown at the bottom right is the series of myofibrils that are found inside of an individual myofiber. Be careful with the nomenclature. Myofiber or muscle fiber refers to the cell itself and all of its constituent parts. The myofibrils are the proteinaceous structures that contain a repeated series of individual units known as sarcomeres that are responsible for contraction, and they are found within the myofiber or the cell. So the myofibrils are made up of a series of repeated iterative proteins that ultimately make up a series of sarcomeres, and that's shown in this slide. These repeated series of proteins are comprised of thick and thin filaments that are held in place by discs. The border of the sarcomere is the Z disc. There's a Z disc on either side, and the Z disc will anchor the thin filaments. The M discs are found between the Z discs, and the M discs will anchor the thick filaments. The thick filaments and thin filaments partially overlap, and this gives us a banded appearance, sometimes referred to as a striated or striped appearance. These bands all have individual names that correspond to those individual bands. So for example, the band corresponding to the lack of overlap of thick and thin filaments surrounding the Z disc is known as the I band. The similar band describing the lack of overlap where only thick filaments are found surrounding the M line is known as the H band. The A band refers to the total extent of the thick filaments, which has both overlapping and non-overlapping portions. Surrounding these myofibrils is a convoluted network of the sarcoplasmic reticulum, which is an organelle which contains large concentrations of calcium. And in skeletal muscle fiber, the main function of the sarcoplasmic reticulum is to be a storehouse for calcium. There are also many mitochondria because of the highly energetic processes involved in muscular contraction. The sarcolemma simply refers to the the membrane around this muscle fiber. And the T-tubule has an interesting role. Because these muscle cells are so enormous, it is difficult to get the signal that occurs on the surface of the muscle cell down to the portions of the contractile apparatus that require the signal for contraction to occur. Therefore, the T-tubules are deep invaginations of the membrane that allow the action potential to traverse and to go deep into the cell in order to allow the signal transduction to occur. This is a requirement because of the mechanical interaction between the sodium channels on the membrane and the ryanodine receptor on the sarcoplasmic reticulum, which causes the release of calcium, which triggers contraction. Shown in the next slide is a more diagrammatic version of the sarcomere. Here we see the Z lines, which are the borders of the sarcomeres, and the M line in the middle, which is the midpoint of the sarcomere, which anchors the thick filaments. The A band corresponds to all of the thick filaments, both with overlap and without overlap. And the I band, though not shown here, shown in the previous slide, represents the non-overlapping regions surrounding the Z lines or the Z discs. It's important to recognize, although these are shown as lines, such as the Z line, in 3 dimensions, these are really more properly referred to as discs. The next slide illustrates in more detail the thick and thin filaments. The thick filament is made up of myosin, and individual myosin monomers have a long, straight portion and then an angled, shorter portion coming off of them. In many ways, they resemble a hockey stick, such that the thick filament is made of the regular organization of many of these hockey sticks put together, with the heads hanging off of the hockey sticks pointing out along all directions radially around the thick filament. The thin filament is made up of 3 different proteins. They're primarily made up of actin. Actin is a globular molecule, and this arrangement of many molecules of actin gives it the length to comprise the thin filament. Very importantly, actin has binding sites on it for myosin, and the affinity for actin to myosin is extremely high. They will bind during contraction, but that binding has to be regulated, and that regulation occurs via a protein shown here known as tropomyosin. Tropomyosin will block the binding sites for myosin on actin. Now, that blockade has to be regulated, and that regulation occurs via troponin. Troponin is a molecule that, when activated by calcium, will move tropomyosin out of the way to reveal the binding sites on actin for myosin, and that will allow contraction to move forward. Troponin also happens to be part of a large superfamily of calcium-binding proteins. There are many types of proteins that will bind calcium. A few of them are shown here. Troponin, as shown here, with troponin C, calmodulin, calbindin. There are many others, but they are sensors for calcium, and when they bind calcium, this will alter their conformation. The next slide illustrates the sequence of events that occurs during muscular contraction. At the top is shown the thick and thin filament at rest. There's no binding between actin and myosin. When contraction is signaled via an increase in intracellular calcium, that calcium will bind to the calcium binding portion of troponin. That will cause a conformational shift such that troponin will move tropomyosin out of the way. Tropomyosin will allow the binding of actin to myosin. When actin and myosin bind, ADP plus an inorganic phosphate will bind to the myosin head. The myosin head, as will be described, has been energized by the previous hydrolysis of ATP, such that when this energized myosin head binds to actin, the act of binding, once that binding occurs, will cause a conformational shift such that the myosin will go back to its non-energized state, or its acute angle state, and in doing so will pull the actin over itself. That will cause the whole filament, the whole thin filament, to slide over the thick filament. In the process of doing so, the myosin will also release ADP. At this point, the actin is still bound to the myosin, and in order for the actin and myosin to be released, this requires a new molecule of ATP to enter to cause the separation of actin from myosin, and that's shown in the transition arrow C. Now at this point, we've not hydrolyzed any ATP, and no energy has gone into this process. Now is when the energy is utilized. ATP will hydrolyze itself. It will release a phosphate group, and in the process, that energy is used to put myosin back into a high energy state. That is, it adopts a different configuration, no longer this acute angle configuration, and is now ready to bind actin again. This process will continue as long as calcium levels are elevated above a certain threshold. This whole process is referred to as the sliding filament hypothesis. So we have the binding of actin to myosin and ADP dissociation, which induce a conformational shift of myosin amounting to about 10 nanometers. In this acute angle conformation, ATP will bind this myosin portion of the actin-myosin conglomerate. That will cause dissociation of the myosin head from the actin-myosin complex. ATP will hydrolyze. That will release energy. That will shift myosin back to its energized position. And as mentioned before, this whole cycle will repeat until calcium levels drop below 10 to the negative 7 molar. Interestingly, this mechanism explains the well-described phenomenon of rigor mortis. Rigor mortis is stiffening of the muscle that occurs after death. This is due to two factors. Number one, the lack of ATP production after death causes intracellular calcium levels to rise because ATP is not available to power the active transport processes that pump calcium out of the cytosol. Second, without ATP, there is no ability for actin and myosin to dissociate. Recall that the role of ATP, or one role of ATP, is to cause actin and myosin to dissociate. So we have both high levels of calcium triggering contraction and low levels of ATP not allowing relaxation, which causes the stiffening of muscle, and this stiffening will remain until that muscle is simply broken down. The next slide illustrates which bands change during contraction. This is important because it's a common source of USMLE questions to quiz you about your knowledge about the physiology of the sarcomere. So shown at the top is the sarcomere at rest. When muscle contracts, the non-overlapping portions get smaller. So for example, the I band, which is a non-overlapping portion around the Z disc, and the H band, which is a non-overlapping portion around the M line, both get smaller during contraction. The A band, which has no relationship to the overlap, it simply is the length of the thick filament, does not change during contraction. So as mentioned earlier, the ryanodine receptor plays an important role in the release of calcium from the sarcoplasmic reticulum. The ryanodine receptor is a complex channel that allows the release of calcium from the sarcoplasmic reticulum into the cytosol, and it's gating. The process by which it gets opened is also complex. The opening of this channel is triggered by the mechanical movement of the sodium channels on the plasma membrane of these muscle fibers, such that when an action potential fires on these muscle fibers and that opens up sodium channels, those sodium channels are physically linked to the ryanodine receptor, which will cause the release of calcium into the cytosol. This is the reason why one needs T-tubules, so that the action potential can occur physically adjacent to these ryanodine receptors. Note that this is different than in cardiac muscle. In cardiac muscle, you also have ryanodine receptors, but they're gated in a different manner. They cause release of calcium via an influx of calcium from the extracellular space. This is referred to as calcium-induced calcium release, and this is a phenomenon that occurs in cardiac but not skeletal muscle. At this point, we're ready to discuss the dynamics of muscle contraction. When a muscle is stimulated to contract, one will observe a twitch. But the duration of the twitch is substantially longer than the action potential that caused that. Because of this, one can observe temporal summation. That's shown in the top figure here, number 2. as multiple stimuli are given in rapid succession, because the twitch is slower than the action potential that caused it, those twitches, the contractions, can build on each other, and eventually one can achieve a fused contraction, which is known as tetanus. And that's shown at the top right as well as in the bottom right. Clinical vignette. You have a 46-year-old female who presents to your office with 4 to 5 weeks of shortness of breath and muscle aches. You examine her and you find that she has a peculiar rash shown here on her upper chest. On exam, she has mild proximal muscle weakness and appears to have diffuse erythema over sun-exposed areas. Her total creatine kinase level is elevated. What is your diagnosis? Polymyalgia rheumatica, Lambert-Eaton myasthenic syndrome, Guillain-Barré syndrome, myasthenia gravis, or dermatomyositis. It's important to put together some of the key clinical features here. We have a patient who has proximal muscle weakness, and recall in a previous discussion that proximal muscle weakness is almost always found associated with diseases of muscle or diseases of the neuromuscular junction. The other very important piece of information here is that the patient has a high level of creatine kinase. High levels of creatine kinase are found in the setting of diseases that destroy muscle, primary diseases of muscle. If we look at the choices given here, polymyalgia rheumatica, Lambert-Eaton myasthenic syndrome, Guillain-Barré, and myasthenia gravis, none of these are primary diseases of muscle. Polymyalgia rheumatica is not very well understood, but it's likely not a disease of muscle, and it certainly is not associated with a high level of creatine kinase. Dermatomyositis is an inflammatory condition that affects muscle and also affects other organ systems such as the skin, and that gives this classic sun-exposed erythematous rash found in this disease process. So the correct answer here is E. Next clinical vignette. A 5-year-old boy is initially brought in because he's not able to keep up with the other kids in his kindergarten. Over time, he develops a waddling gait. His gait worsens, and by age 10, he needs knee braces for stabilization, and by 12, he's in a wheelchair. On a physical examination, he has diffuse muscular atrophy with enlarged calves. So this patient has Duchenne's muscular dystrophy. This is the X-linked loss of dystrophin, and as shown in the upper right, dystrophin is a molecule which links actin with the extracellular matrix, and this linkage occurs through a complex of proteins associated with the cell membrane. Loss of dystrophin will cause progressive muscle wasting and ultimately death by the teenage years. This is a very classic presentation of Duchenne's muscular dystrophy, which causes proximal muscle weakness and a progressive loss of strength over time. The need for the knee braces is part of the proximal muscle weakness that I described earlier. The other clinical manifestations here are a progressive cardiomyopathy, which is seen in these patients. TREP, muscular TREP. So TREP is another phenomenon related to the dynamics of muscular contraction. So shown here on the bottom right is the sequence of contractile strengths observed after multiple contractions of a muscle are initiated. So on the graph as shown, there's an initial contraction, which is a particular tension, and then when the muscle is stimulated again, the muscle contracts with slightly more tension, with a progressive increase in the amount of contractile force with the subsequent contractions over time. The phenomenon shown here is not temporal summation. The reason it's not temporal summation is because the contraction returns to baseline after each stimulus. Instead, this is a different phenomenon known as TREP, or muscular TREP. The underlying mechanism for this is not entirely understood, although it is clear that there are probably two different contributing factors. One factor is calcium. Every time a muscle contracts, calcium gets released not only from the sarcoplasmic reticulum, but there's also calcium which gets into the cell from the extracellular space. After contraction, this extracellular derived calcium is sequestered back into the sarcoplasmic reticulum such that more calcium is available for the subsequent contraction. As this process continues, the contraction gets stronger and stronger until a threshold is reached. The other phenomenon that's likely responsible for the increase in muscular strength is the increase in the activity of all of the proteins involved because of the heat generated by muscular contraction. Whenever ATP splits and releases energy, only a portion is used to power the muscle contracting. The rest of this is released as heat. And as all of these enzymes are warmed up, they all perform more efficiently, and the contractile apparatus all these sequences occur more rapidly, and this causes more synchronized and therefore stronger contractions. This is the rationale why it is recommended that people warm up before working out, not only for safety reasons, but also in order to increase performance. In the next slide, we're going to introduce the force, velocity, and length relationship. When a muscle is contracted, its strength of contraction will depend on the baseline length of that muscle. This is known as preload dependence, and this is illustrated on the graph on the right. If one were to take a muscle, as shown in this figure, fix its length, and induce it to contract, one will get a particular amount of stress. And if one were to increase the length of this muscle sequentially, one will find that the strength produced will vary over length. And that's shown on the top figure with the brown circles. And if one were to look at this function, it looks quite complex. It looks like there are two peaks. There's an early peak and then a later peak. And the reason that there are two peaks is because there are two separate processes that are responsible for the tension produced by this muscle. One process is all of the active physiology that we've been discussing. Actin and myosin interacting, the pulling of the thin filament over the thick filament, etc. The other process which is contributing to the tension produced by this muscle is simply the passive elastic pullback produced by this muscle. Any elastic molecule, or any elastic substance will pull back. If we were to put a rubber band into this experiment and we pulled on it, it would also pull back. So if we wanted to separate these two different effects, these two different processes, the active and the passive processes, we could repeat the experiment, put this muscle back on the in his apparatus, and rather than stimulating it to contract, we can just stretch it and see how much tension is produced by that muscle. So if we do that experiment, we get the results shown with the blue circles on the bottom. One gets an exponential rise in the amount of stress produced by this muscle when one simply pulls on it rather than one contracts it actively using the stimulator as shown. If you were to to subtract the passive curve from the total curve, one will obtain this curve in the color deep red or brown, this upside down U shape, and this corresponds to the active tension produced by the muscle. This active tension is produced by all the physiological processes that we've been discussing. Again, actin overlapping with myosin, the two of them interacting, and the sliding filament hypothesis creating tension. The reason that this relationship holds, and that is the reason why there is an upside down U shape such that there is a middle range of maximum contractile force and an upper and a lower range with less contractile force, is because of the orientation of actin versus myosin in these various lengths. So as shown on this figure, if you look at the point all the way to the bottom right, here is where the muscle has its longest length. And here is where there is the least amount of overlap between actin and myosin. When one tries to contract the muscle, when the muscle is stretched out like this, you're not going to get a strong contraction because the overlap between actin and myosin is poor. Conversely, all the way to the left of this figure, when the sarcomere has been artificially shortened and there is not good overlap or efficient overlap between actin and myosin, if anything, there's steric hindrance amongst the actin molecules, one also does not achieve optimal contraction. In the middle range, where there is good actin and myosin overlap, here you have maximal contractile force of the muscle. And this is the reason why there are particular preloads or lengths where one can achieve maximum muscular contraction. Another important relationship is the relationship between load and power. Power of a muscle is computed by knowing how rapidly it contracts and against what force it's contracting. One can do There's some terminology here we should be familiar with. So the preload on a muscle then is the load on a muscle before it contracts. And often preload is really referred to as a length. When you talk about the load on a muscle, whenever you load a muscle, it will alter its length. So the preload in many ways simply refers to what is the length of the muscle before it contracts. And as we learned 2 slides ago, this will have a major implication on how much tension that muscle can produce. The afterload is the load that the muscle pushes against as contraction begins. Isotonic refers to any situation where the load is constant. If one were to look at the previous slide at the load power curve, that was a curve that was generated using a series of isotonic contractions. Isometric refers to a situation when contraction is occurring at a constant length. If one were to look back at the length tension curve, that curve was generated using a series of isometric contractions. Each contraction was conducted at an individual length, and then all of those lengths were put together to construct that curve. Power refers to work per unit time, and functionally one can compute the power by looking at the force, which is the same thing as the load, times the velocity. That's what we did in the previous slide. We multiplied the load times velocity to compute the power. And then finally, stress. When one talks about the output of a muscle, one can simply talk about the force of that muscle, but it's not fair to compare the force produced by a large muscle like the quadriceps to the force produced by a smaller muscle like the extensor indicis. So if you were to divide by the cross-sectional area of the muscle, one will have a normalized metric of force, which is referred to as stress. And one can now appropriately compare the force or the stress or the output produced by a muscle under various circumstances. Question 20. A 55-year-old man presents with an acute myocardial infarction and congestive heart failure. In addition to his acute therapy for the plaque rupture, he's given intravenous Lasix or furosemide. Why is this given? A, to decrease preload, B, to increase preload, C, to prevent renal failure, D, to clear the angiogram contrast from his system, or E, to reduce potassium load. So the answer here is to decrease preload. When somebody has congestive heart failure, they have functional fluid overload, and if one were to look back at our length-tension relationship, you'll find that those cardiac myocytes are way on the right-hand side of that curve, and that is their preload is too high. They are in a configuration where the actin and myosin have poor overlap, and by decreasing preload, one can put those sarcomeres into a configuration where they have more optimum overlap and hence greater cardiac output. So dropping preload with a potent diuretic is very commonly done in the setting of acute congestive heart failure. During an acute stroke, blood pressure is often elevated. Patients with coronary disease tend to, quote, rule in, that is, elevated cardiac enzymes are found in their serum. Why? A, the, quote, cardiac enzymes are also found in the brain, and what is found in the serum reflects brain damage. B, the elevated blood pressure increases preload on the heart. C, the elevated blood pressure decreases preload on the heart. D, the elevated blood pressure decreases afterload on the heart. Or E, the elevated blood pressure increases afterload on the heart. The correct answer here is E. The elevated blood pressure will increase the afterload on the heart. In the setting of a stroke, the brain will cause the blood pressure to be elevated as part. This concludes our discussion of the physiology of skeletal muscle.